For decades, oncology textbooks have told us a fairly comforting lie about how cancer works.
Yeah, that it's a slow, linear process.
Exactly. The standard model has always been that a tumor takes years or sometimes decades to figure out how to spread.
Right. The assumption was that metastasis is just this late-stage accident.
Yeah, a final deadly mutation that a tumor only unlocks after it has grown massive and, well, chaotic.
But what if the tumor has a getaway car running before it even commits the crime?
Welcome to the deep dive. Today we are looking at a brand new, just fundamentally paradigm-shifting study published in Nature in April 2026.
And we're focusing specifically on colorectal cancer here.
Right. Our mission today is to dissect a really specific question with you. How, and critically, exactly when does cancer learn to spread?
Because if we look at the source material here, researchers have discovered something terrifying.
The capacity for metastasis is actually baked into the tumor at the microscopic, very earliest moments of invasion.
Which completely upends the traditional linear progression model we just talked about.
Yeah.
If the cellular machinery for metastasis is present at the absolute inception of the disease, we're looking at a deeply coordinated biological event.
Not just a random late-stage mutation.
Exactly. And the clinical implications of this cannot be overstated for you or anyone listening. Metastasis is responsible for the vast majority of cancer-related deaths.
So if we misunderstand the timeline of when a tumor becomes metastatic, our entire therapeutic window is just misaligned.
It is. This paper forces us to examine the very border where a benign growth transitions into a lethal invasive threat.
Okay, let's unpack the methodology here, because the researchers went straight to the scene of the crime to figure this out.
They did. They focused exclusively on early-stage colorectal tumors.
Specifically the T1 stage, right?
Right.
And just for context for you listening, the inner lining of the colon has a very thin boundary wall of muscle. It's called the muscularis mucosae.
A bit of a mouthful.
Yeah, biology loves its Latin. But in a T1 stage tumor, the cancer has just barely breached that wall.
It's just pushed into the underlying layer, the submucosal.
So it's the absolute earliest definition of invasive cancer.
Right. And to understand what is happening at that exact boundary, bulk RNA sequencing isn't going to cut it.
Because if you take a tissue sample and grind it up to sequence the RNA, you lose all the spatial context.
Exactly. It's a biological smoothie. You don't know where any of the cells actually came from in relation to each other.
So instead, the team utilized spatial transcriptomics.
Specifically, the nanostring GeoMx platform. They use this to map 19 of these early T1 tumors.
And just so we're all clear on the mechanics of spatial transcriptomics, because it's such a beautiful technology, they're essentially laying the tissue on a slide.
Right, yes, and tagging the RNA transcripts with barcodes.
Right, barcodes attached to UV cleavable linkers. And then they shine a laser only on highly specific microscopic polygons of the tumor.
It's incredibly precise.
So they can selectively clip and sequence the barcodes from the normal tissue, the dense tumor core, and crucially, the invasive front.
Which is the exact leading edge where the cancer is pushing into healthy tissue.
Right. And the precision of that technique is what yielded this massive, explosive finding.
At the invasive front, they identified a distinct subpopulation of cancer cells.
And these cells were exhibiting what they call profound oncofetal plasticity.
Oncofetal plasticity.
Yeah, we hear that term thrown around a bit in developmental biology, but seeing it in a tiny early-stage tumor is alarming. What exactly are these cells doing?
Well, they are essentially abandoning their mature differentiated states.
Like hitting a reset button.
Exactly. A standard colon cell has a highly specific job, but these cells at the invasive front were upregulating gene expression programs normally restricted to embryonic development.
Or extreme tissue regeneration.
Right. We are talking about genes like LAMC2, MMP7, and ANXA1.
Let's break those down because the specific genes really tell the story of the crime here.
They do.
LAMC2 produces a subunit of laminin. Right.
Right, which provides the traction cells need to migrate.
And then MMP7 codes for matrix metalloproteinases.
Which are literally the molecular scissors a cell uses to chew through the extracellular matrix.
Wow. And ANXA1 alters membrane dynamics,
making the cell highly adaptable and squishy.
Putting that all together, you have a cell that has suddenly acquired the ability to detach, dissolve the tissue around it, and physically deform itself.
Just to squeeze into blood vessels. It has rebooted its software to become a highly mobile, squishy survivalist.
But wait, if I'm looking at this critically, the tumor has barely broken through the first layer of muscle.
Right.
It's a localized microscopic breach.
Yeah.
Why is it activating a massive systemic survival program so early?
It seems like overkill, right?
Yeah, it feels like gearing up for a cross-country bank heist just to steal a candy bar.
That disproportionate response is exactly what caught the researchers' attention.
I mean, it makes no sense under the old model.
No, it doesn't. And they didn't just rely on those initial 19 tumors.
Right, they had to prove it wasn't a fluke.
Exactly. They validated this oncofetal signature across a massive tissue microarray of 232 different T1 colorectal cancer specimens.
232.
Huh.
So the presence of these metastasis-ready cells wasn't a rare anomaly.
No, it was ubiquitous. Almost every single early tumor had them lingering right at the invasive front.
Okay, let's do a quick recap for you listening, because if I'm putting myself in your shoes right now, there is a glaring paradox here.
A huge bottleneck.
Yeah. We've established that almost all early T1 tumors have these highly dangerous, shape-shifting oncofetal cells at their leading edge.
Right.
But clinically, we know that not every patient with a T1 tumor immediately develops metastatic cancer.
Thank goodness.
Exactly. So if the getaway cars are all gassed up and running, why don't they all drive away?
It's the ultimate biological bottleneck. The researchers recognized this paradox immediately and turned to long-term clinical data.
They tracked the 5-year follow-ups for these patients.
And they found that despite having these high relapse oncofetal cell signatures, roughly half of the T1 tumors never metastasized.
Which tells us that oncofeplasticity is necessary for metastasis, but it is demonstrably not sufficient.
Yes. The cancer cells have the tools to migrate, but something is stopping them from actually executing the spread.
So to isolate that missing variable, the researchers compared the transcriptomes of the T1 tumors that eventually metastasize against the ones that remain localized.
And the defining difference wasn't the migration tools.
No.
It was immune evasion. The successful metastatic tumors had heavily downregulated their immune-related expression programs.
Oh, wow. This is where it gets incredibly insidious.
It really does.
Downregulating immune programs usually means the tumor is dropping its antigen presentation, like shutting down MHC class 1 molecules.
Exactly. So the tumor cell has the map and the vehicle to spread.
But if it hasn't figured out how to turn invisible to the body's immune surveillance, the immune cops, basically.
Right. The circulating CD8 positive T cells.
Yeah, they'll just recognize it as foreign debris and destroy it in the bloodstream.
So the invisibility cloak is the required secondary skill.
Without it, the getaway car just gets pulled over immediately.
Perfect analogy. It illustrates how demanding the metastatic cascade is.
The cells must endure a gauntlet of selective pressures.
And merely being plastic isn't enough to survive the immune system's checkpoints.
Okay, so we've established what these cells look like and why only some of them succeed. But I want to pivot to the fundamental origin of these oncofetal getaway drivers.
Because this is where the paper really challenges foundational oncology.
Where do they come from?
Well, the classical assumption is that cancer is a disease of mutated DNA.
Right.
Therefore, a new cellular behavior implies a new genetic mutation.
Exactly. If the cells at the invasive front are behaving wildly differently than the lazy, just sort of proliferating cells in the tumor core, you would assume they acquired a new driver mutation.
Right, that's the logical leap.
So to test this, the team built a multi-regional organoid biobank. And the fact that they did this across multiple regions of the same patient's tumor is brilliant.
Oh, it's an incredibly labor-intensive process.
How many biopsies did they do?
They took 73 individual punch biopsies from 16 patients.
Wow.
And for every single patient, they grew 3D organoid cultures from the healthy adjacent tissue, the tumor core, and that critical invasive front.
So by culturing these regions separately, they basically created living in vitro avatars of the tumor's spatial geography.
Exactly.
And then they threw whole genome sequencing at all of them.
Right, because we know early colorectal cancer relies on classic pathway mutations.
Like APC to disrupt WANT signaling.
Or TP53 to knock out the genome's spell checker.
And KRAS to lock the cell into a constant growth loop.
So the crucial question was, what extra mutations did the invasive front acquire to become oncofetal?
Let me guess. The sequencing results revealed absolutely 0 additional driver mutations at the invasive front.
0. Absolutely none.
That is wild.
The genomic architecture of the highly plastic metastasis-ready cells was completely indistinguishable from the rigid, standard cancer cells sitting lazily in the core of the tumor.
Hold on. Cancer is fundamentally driven by its genetics.
Usually, yes.
Are you telling me these wildly different shape-shifting cells have the exact same DNA as the core cells.
The exact same.
How is that biologically possible? If the genome isn't driving this lethal transformation, what is?
It conclusively proves that this specific step in tumor evolution is not intrinsic to the cancer cell itself.
So the difference isn't genomic.
No, it's transcriptomic and epigenetic. The cancer cell isn't changing its own instruction manual.
Oh, I see. Instead, the surrounding environment is selectively reading different chapters of that manual.
Precisely. The trigger lies entirely in the tumor microenvironment, or the TME.
The neighborhood is corrupting the cell.
Yes.
It's not about who the cancer cell is. It's about who the cancer cell is talking to.
Exactly.
So we have to map the neighborhood to find the culprit whispering these toxic instructions.
And to do that, the researchers deployed single-cell RNA sequencing to dissect the cellular composition of the microenvironment.
And their focus immediately narrowed in on fibroblasts.
Right, fibroblasts.
Fibroblasts are the structural architects of our tissues. They manage the extracellular matrix. They handle wound healing.
They are the builders.
Now, in a healthy intestinal crypt, which are the little valleys in the gut lining where cells regenerate, fibroblast populations are strictly zoned, right?
Strictly zoned, like city planning.
Okay, so how does that zoning work?
At the top of the intestinal crypts, you have fibroblasts, known as telecytes.
Telecytes, okay.
Their main job is to secrete inhibitors that keep cell growth in check.
So they're the brakes.
Exactly. But down at the very base of the crypt, you have a completely different subpopulation called trophocytes.
And trophocytes are crucial because they maintain the stem cell niche.
Right. They secrete the exact growth factors that keep the healthy intestinal stem cells happy and regenerative.
The gas pedal. But when the researchers looked at the spatial distribution of these fibroblasts in the T1 tumors, the zoning laws were completely corrupted.
Completely thrown out the window.
Deep inside the tumor core, they found normal telecytes.
But out at the invasive front, right where those dangerous oncofetal cancer cells were congregating.
Let me guess. They found trophocytes.
Sort of. They found massive clusters of trophocyte-like cancer-associated fibroblasts, or CFs.
The tumor essentially forces a zoning rewrite.
Yes. These cancer-associated fibroblasts were highly expressing markers like FAP.
Fibroblast activation protein.
Right, and SFRP2.
So it's a complete architectural hijacking.
It really is.
It's like an invading army doesn't just conquer a city. They force the local architects to build them a custom fortress.
That's exactly what's happening. The invading tumor isn't just pushing into healthy tissue, it is actively recruiting the local fibroblasts.
Turning them into a corrupted version of trophocytes.
And forcing them to build a synthetic stem cell niche right at the border.
But the spatial correlation was undeniable, right? The oncofetal cancer cells and these trophocyte-like CFs were locked together at the invasive front.
They were. But in molecular biology, spatial correlation doesn't satisfy the burden of proof.
Right, you can't just say they're in the same room, so one must have killed the other.
Exactly. The researchers had to prove that the CFs were actively causing the cancer cells to change.
And this is where the 3D organoid cultures come back into play, which I think is just the most elegant experiment in the entire paper.
It's fantastic.
They set up co-cultures in matrigel, which mimics the extracellular matrix.
Right.
They took those core non-plastic tumor organoids and then deliberately introduced the corrupted fibroblasts into the dish.
And the results were striking. When cultured alone, the tumor organoids remained relatively standard.
Rigid and lazy.
Yes. But the moment the trophocyte-like CFs were introduced, the cancer cells underwent a radical shift.
The fibroblasts actively induced the oncofetal state in the tumor cells.
It was a direct, causal cell-to-cell manipulation.
They literally brainwashed the cancer cells. So what is the chemical whisper? How exactly is the fibroblast communicating this command across the extracellular space?
Well, transcriptomic analysis of the CFs revealed they were secreting massive quantities of TGF-beta.
TGF-beta, which is a notorious driver of epithelial to mesenchymal transition in biology.
It is. And furthermore, they heavily expressed an enzyme called PTGS2.
Which is better known as COX2?
Right.
We usually talk about COX2 in the context of inflammation.
Like taking ibuprofen to block it.
Exactly. It's the rate-limiting enzyme for producing prostaglandins.
Precisely. Prostaglandins are potent lipid autocoids that dramatically influence cellular plasticity and inflammation.
So the hypothesis was that the combination of TGF-beta and prostaglandins was the specific key unlocking the oncofetal program.
Yes. And to prove that definitively, they used CRISPR.
Oh, the methodology here is just phenomenal.
It really is beautiful.
For you listening, they used homology-directed repair to insert a fluorescent tag directly into the tumor organoids.
Specifically, they targeted the EMP1 gene locus.
EMP1 stands for epithelial membrane protein 1.
Right, and it is a massive hallmark of that dangerous high-relapse oncofetal state.
So by splicing a fluorescent reporter right next to the EMP1 promoter, they created a real-time visual alarm system.
Yes. If the cell turns on the oncofetal program, the EMP1 gene transcribes.
And the cell physically glows green under the microscope.
They built a security camera for metastasis.
That is so cool.
And when they bathed those pure tumor organoids in just recombinant TGF-beta and prostaglandins.
Without even the fibroblasts present.
Right, just the chemicals, the dishes lit up, the cells glowed.
It is ironclad proof. The specific chemical cocktail of TGF-beta and prostaglandins is the exact trigger.
The exact trigger that forces a static tumor cell to regress into a shape-shifting metastasis-ready state.
It beautifully connects the macro behavior of the tumor to the micro signaling of the microenvironment.
Because the cancer cell isn't mutating to gain new abilities.
No, it is simply responding to the inflammatory regenerative cues being blasted at it by corrupted local fibroblasts.
Exactly.
Which brings us to the final and perhaps most suspenseful part of the deep dive. Let's do another quick recap.
Sure.
We have identified the players, the oncofetal tumor cells and the corrupted CFs.
We know the weapon, TGF-beta and prostaglandins.
We even know the location, the invasive front. But chronologically, when does this unholy alliance actually form?
This is the critical question. To capture the exact moment of inception, the researchers utilized an even more advanced spatial technology called CosmX.
CosmX. Unlike GeoMX, which profiles small regional polygons, CosmX provides true single-cell subcellular resolution.
Right. It allows you to look at the exact transcript of a single cell and its immediate touching neighbors.
So they applied this to 11 highly specific early-stage specimens to create what they called a pseudo-timeline.
Because you obviously can't watch a tumor grow in real time inside a human.
No. You have to look at static slides representing different chronological microstages of invasion.
And the timeline was broken down into 3 critical stages.
Okay, lay them out for us.
First is the intramucosal carcinoma. This is the stage just before the tumor touches the muscular wall.
The wall being the muscularis mucosa.
Right. Second is the T1-SM1 stage, which is the exact microsecond the tumor physically breaches that wall.
Okay, and the third?
Third is T1-SM3, where the tumor has firmly established itself deep in the submucosa.
Watching this data unfold is like watching a slow-motion collision. At the first stage, the intramucosal carcinoma, before the wall is breached, what is happening in the submucosa?
Absolutely nothing out of the ordinary.
Nothing.
The submucosa is completely calm. The tissue-resident trophocytes are just sitting there, maintaining the normal tissue.
And a tumor cell's above the wall.
They're proliferating, sure, but they show 0 signs of oncofetal plasticity.
But the absolute millisecond, the physical barrier of the muscularis mucosae is compromised at the T1-SM1 stage.
The entire ecosystem violently shifts.
It's immediate.
It is a simultaneous, instantaneous reaction. The sheer physical presence of the tumor cells breaking into the submucosa immediately forces the resting trophocytes to transition.
Into those corrupted trophocyte-like CCAFs.
Yes. And the very second those CHAFs transition, they flood the localized area with TGF-beta and prostaglandins,
which instantly forces the breaching tumor cells into the oncofetal state.
The timeline is instantaneous. The tumor doesn't slowly adapt to its new environment over years.
The moment the border falls, the collision between the tumor and the naive submucosa sparks an immediate chemical fire.
The capacity to metastasize is birthed at the very first moment of invasion.
It completely shatters the notion that we have years to intercept a slow-learning tumor.
It does. The invasive front is a dynamic, real-time chemical reactor.
The tumor weaponizes the body's own wound-healing response against it the second it causes tissue damage.
This requires a massive cognitive shift for anyone in the medical field.
And, frankly, anyone trying to understand the nature of this disease. For decades, we have poured billions of dollars into therapies designed exclusively to hunt and kill mutated cancer cells.
Right. Precision oncology.
But this research explicitly proves that the tumor's most lethal asset, its ability to spread, isn't even a genetic trait of the cancer itself.
It's a behavioral response dictated by the neighborhood.
If the microenvironment dictates the tumor's deadliness, then our therapeutic targets have to expand.
Absolutely. Targeting the cancer cell's genome with precision oncology is only half the battle.
Because if we don't address the fibroblasts whispering these toxic survival commands, the remaining tumor cells will just keep being pushed into a metastatic state.
It opens up an entirely new frontier of treatment.
What if instead of just carpet bombing the tumor, we could chemically silence the CFs?
Right. If we can block the TGF-beta receptors.
Or aggressively inhibit the COX2 pathway, right at the site of early invasion.
Could we cut off the chemical whispers?
The theoretical application is profound. If you neutralize the microenvironment, you strip the tumor of its plasticity.
You trap the cancer in its rigid, predictable state,
making it vastly more susceptible to traditional immune clearance or surgical removal.
You turn a systemic, shape-shifting threat back into a manageable, localized mass.
As we wrap up this deep dive, I want to leave you with a philosophical, almost provocative thought that extends far beyond oncology.
Oh.
We've spent this entire time dissecting the dark, destructive brilliance of this process. The tumor is hijacking the human body's most guarded secrets.
The fetal blueprints of development.
And the pathways of extreme tissue regeneration, just to survive.
It's using the biology of life to drive the pathology of death.
But think about the sheer power of the mechanism itself. A simple combination of local cellular signaling, TGF-beta and prostaglandins, is enough to force mature, biologically locked adult cells to regress.
To regress into a highly plastic, flexible, regenerative state.
Cancer has mapped the exact chemical code required to turn back the biological clock of a cell.
Which is the fundamental holy grail of regenerative medicine.
Exactly. If science can fully decode the exact signaling cascade that cancer uses to force adult cells to become plastic and embryonic.
Could we harness that exact same localized chemical whisper for healing?
Yes. If we can master the CAF's playbook, could we intentionally induce that same plasticity in a damaged human heart after a massive myocardial infarction?
Wow.
Could we use this exact inflammatory signaling to force a severed spinal cord to regenerate its neuronal connections?
It is the ultimate paradox.
Right.
The precise microenvironmental hijacking that makes cancer so impossibly lethal might inadvertently hand us the instruction manual for limitless human regeneration.
The landscape of early cancer is terrifyingly complex, filled with corrupted structural workers, spatial zones, and chemical brainwashing.
But by mapping that chaos, we aren't just learning how to trap a tumor.
We might be uncovering the fundamental blueprints to heal everything else. Something to think about as you go about your day. Until next time.